Hi everyone. I hope I am audible. So let me just check. Yes. Okay. So let's get started. So let's have a look at the kidney structure today, and also let's see how the arrangement of nephron, that is uriniferous tubule, a renal tubule or a kidney tubule, so arrangement in the cortex region and the medulla region, let's have a look at that as well. Okay, and let's have a look at the blood supply which is done to the kidney. So we will cover those topics today. Yes, welcome to the class. So before I start, let me introduce myself. I am Vani Kari, biology educator at Unacademy. I am an engineer with bachelor's and master's degree in biotechnology and a B.Ed degree. Also a founder of a startup where I grow microgreens by vertical farming technology. Okay, so join with us in Telegram. So for homework and class notes, you can join the channel Vani Kari ICSE class 9 and 10, and Telegram channel, that is Unacademy ICSE and ISC both. So this link you will find in the description box. Okay, so go to the description box and click on the link to join this channel, and you will be getting the updates regarding the classes there as well. Okay, also we are conducting a special classes on Unacademy platform, so those are absolutely free. And what exactly happens in those classes? It is a live real-time interaction using the chat and emojis. Okay, so regularly, like you will have a live classes where you can attend the classes for different subjects. Okay, so there also the chat option is available and emojis are also available to react. So let's say discussion session is going on, so you interact with the other learners or you want to interact with the educator, so all that is possible using this one. And other than that, so since lot of comments keep on coming, so there is a chance that your question might get lost. So that is why there is a separate tab, a question tab is there, where you can type in your question, attach an image, send it to the educator during a live class, and they'll be getting a notification and they'll be clearing your doubts. Okay, also we are conducting a quiz. Okay, so that is a poll war for you all. So basically the question and options will be available on the screen. Okay, so we would add that, and then you will get the options like A, B, C, D to choose between. Okay, so basically we are conducting a poll war there or a quiz there, and so with that you will be getting your ranks also. So we will be sharing the leaderboard so that you can compete with your friends as well. Okay, so you will know your ranks, and it can give up to 10 ranks it is giving. Okay, so you can have a look at your ranks also, and there is a chance that you can compete with your friends as well, because lot of students will be there from different states. Okay, so you get to compete between them. All right, so Unacademy subscription feature, so this is about the plus classes that we are conducting. So here in this case, so these are not the free classes, so free classes are the special classes, and here in this case you can learn live from the comfort of your home. So this is applicable for both as well. Unlimited access to all the courses. There are different courses which are launched by different educators. So if you take the plus subscription, so you will have access to all of these courses. Okay, so top educators of India are going to take the class in one platform, that is Unacademy platform. Then there is regular doubt clearing session. So after about every 3 session, the 4th session which we are conducting, so that is actually the doubt clearing session, or we call it as an answer writing session also. So what exactly is this? So any doubts you have related to the topics that are previously covered, so you can ask your doubt in this particular session. Okay, and let's say you do not have any doubt, so we can conduct a poll war also, or let's say you do not have again any of this, so we can revise also. So this will be completely dedicated for learners itself. Then complete coverage of a syllabus is done, mentorship and guidance is given, study material is given, then the practice test, live test series, batch courses. So depending on your preference. Okay, so if you want to learn in Hindi, so there is a Hindi batch available. If you prefer to learn in English, then there is a English batch available. If you prefer to learn in English batch is also available. So you will have access to all of this. Okay, so then daily practice session, the weekly mock test series, live quizzes, then daily MCQ and subjective test series. So you can take a benefit of all this with the plus subscription. So let's say you want to try like how exactly the plus subscription works. So maybe you can go for 6 months or 9 months, and so if you are planning to complete your class 11 and 12, so you can go with the 24 months, or maybe depending on your convenience you can go for any of these months of subscription. So this one for trying you can go for 3 months also. For class 10 3 months is available, and this is my referral code Vani YT. So with this referral code you will be getting 10% discount on the subscription. Okay, so other than that we have a offer, so mega offer for plus subscribers. Okay, so basically for all the classes, class 9, 10, 11 and 12, so let's say you are buying 12 months or more of a subscription, so you will get a free 2 months extension here. So flat 10% discount on purchasing plus subscription of any duration. So here again valid for first 500 subscribers. And how exactly are you getting flat 10%? So that is with the referral code Vani YT. So you need to enter a referral code, then only you'll get a flat 10% discount. Okay, so don't forget to add a referral code there. So if you have any queries, so you can get in touch with your counselor. So confused about your preparation journey, or have queries about the subscription, or want to know about the latest offer, so you are just one call away. So you can call on this number and again use the referral code Vani YT to get 10% discount on your subscription. Okay, so let's get started. Yes, anybody, what is kidney, what does it do, and how many kidneys are there? So in each kidney how many nephrons are there? Yeah, so how exactly they are arranged, what is the kidney doing, why the blood is going there? Okay, so lots of questions are there. Kidney is it is performing a majority of function. Getting rid of the waste thing is the major function actually, because what happens if this waste product stays in your body? All the tissues are going to get damaged, and if all the tissues are going to get damaged, what will happen? All the tissues are dying. So that is why one need to get rid of these unwanted material. And how are these unwanted materials formed in our body? Yes, anybody? So how exactly these unwanted materials are formed? So that is waste materials what I am talking about. So those are based on your lifestyle. Okay, so lifestyle is the one which is bringing a lot of changes. Okay, so now let's say you are getting exposed to a lot of carbon dioxide that you are consuming. So in that case it is the lung that is going to perform the activity where exhalation is done. Okay, so there is lot of carbon dioxide synthesis that is done. Okay, so this is with respect to lung. So let's talk about kidneys. Okay, so in case of kidneys, what is happening? So what is the waste product, how is it generated? Yes, anybody wants to answer this one? I don't know. This is chat is not available. Anyway, let's have a look at this one. Okay, so in this case, how exactly the waste products are formed? Depending on your diet again. Okay, so if you are consuming more of a protein rich diet, okay, so let's say you are taking a protein rich diet, depending on your lifestyle, so there is a requirement of a protein for all your, like, all your cells require a protein for their formation. So here, so now here let's say this is a cell, okay. So you know that cell will be made of a phospholipid will be there, so there is a phospholipid bilayer like this, it will be arranged, okay. So in between you can find, there are protein blocks like this, okay. So again there is a phospholipid like this, okay. So this will be the phospholipid chain, okay. This is a bilayer which is of plasma membrane, and again you can see integral proteins, okay. So now these integral proteins are responsible for, most of the functions. So they'll allow movement of certain things in and outside, and you know that lot of cells are actually generated. So your body requires protein for cell division, cell formation, then for enzyme formation, because again it is enzymes are proteins, okay. So they are protein in nature. So with respect to that required amount of protein, what is there? Okay, so that will be used up by your body, but extra amount, okay, so excess amount or extra amount of protein, what is there? So this is actually not stored in our body, like you consume, like you like lot of sweets, so you consume lot of different, all the varieties of desserts, you get a chance, okay, you get a chance to eat all of this, like different varieties of desserts. So when you go for it, so carbohydrate part, what is there? So you are consuming excess of it. So you know that carbohydrates are stored in our body, but this is not the case with proteins. Proteins are not getting stored, okay. So excess amount of protein, what is there? So this excess amount of protein itself is converted into urea, and urea is what? It is a waste product, and we need to get rid of this waste product, because what will happen if urea is there? So this is a toxic compound, and we don't want this toxic compound to remain in our body. So our body is trying all its best to protect your kidneys, to protect your different tissues. So there is circulatory system, okay. So this is like the circulatory system, it's not that we can say this is the best system anyway, because all the systems are performing their best actually. So let's have a look at this one. So circulatory system is the one which is carrying this waste product to the kidney, then from kidney it is going where? So from kidney the waste products are filtered out, and it is going out in the form of a urine. So you can see in this picture, okay. So what exactly is happening? So there is a ultrafiltration process that has occurred, so through the kidney. So now you can see this one. So here the blood supply is done. This is actually this movement what you see here. So that is a ultrafiltration process where the filtrated product of a blood, okay, so glomerular filtrate we are receiving at the Bowman's capsule. So let's have a look at that. Okay, so before that, kidneys are bean shaped excretory organs, okay. So these are the bean shaped excretory organs. So functions of kidneys are excretion and osmoregulation, okay. So excretion is done. What exactly is osmoregulation? Okay, so you know that the blood which is entering your kidney, okay, so that is getting filtered. That means liquid part of the blood what is there, so that is going out, in the form of a urine, but not all the liquid, because our kidneys what are there, and mainly the structural units and the functional unit, the nephrons, so they are too clever. So basically reabsorption will occur from certain parts of them through the capillaries again. So in that case the normal concentration of blood is maintained, and excess amount of liquid is the one which is going out in the form of a urine, okay, and with that a more of a urea will be there, okay. And now what osmoregulation meaning is that maintenance of the normal concentration of the blood after the filtration is done. So that is the osmoregulation. So maintaining the osmotic pressure of the blood, so that we call as the osmoregulation. So kidneys help in filtering the blood. blood. Okay, so that is where the filtration is occurring. So blood is getting filtered. So from blood only urine formation is occurring. So it is not something new liquid is created in our body. So blood itself is getting filtered, and this waste product which is there in the blood, so that itself is a urine, but there is no RBC, there is no WBC, there is no platelet. So plasma part with all the unwanted things like waste materials are there. Okay, so here again, only when abnormal condition, whenever a disease is there or infection is there, so in that case we can find blood in the urine. So you might have noticed or you might have heard about this. So blood in the urine, then the infection, pus formation which we can see with respect to certain parts of a excretory system, so that the urine which is formed, so that will be turbid. Okay, so all this can happen with respect to infection, or otherwise urine is basically a filtrate. So filtrate from where? So filtrate we have obtained from the blood itself. Okay, so let's get started. So this is the structure of a kidney. Okay, I mean the nephron. So let's have a look at the kidneys now. So kidneys, when we mention, so these are two vein shaped organs. Yes, remember here location of the kidney, or that is right kidney. So they'll ask you to draw this one, and this one is right kidney. Okay, and this one is left kidney, and you need to notice here that the left kidney is slightly above the right kidney, like so this one is slightly below, whereas the left one is slightly above. So that you need to draw when you are drawing it for your exam. So make sure that you are drawing the right kidney slightly below. Okay, so this one is slightly above and this one is slightly below, so that is for the right kidney. All right, so now these are two bean shaped organs. So what is the length of them? So this is about 10 centimeter long, 6 centimeter wide. Okay, and these are basically located on the urinary bladder. So location, exact location you need to mention, the one which is given in your textbook. Okay, so ICSE evaluation, so we'll look at this itself, whether you have written the exact location or not, whether you have included the key words or not. Key terms will be there, so you need to include them, then you are going to get full marks. Okay, so make sure that you are including all the important words like keywords. Now here, as you can see that, so located on the urinary bladder has to be there. So this is, these are bean shaped structures, so which are present there at the back of the something. So if you randomly, if you are writing on your own, so you are not going to get marks. So exact location has to be written. Okay, so urine is intermittently emptied from the urinary bladder to the outside of the body through the urethra. Now here is the urethra. So through the urethra, when the urinary bladder is almost full, so under the impulse from brain, the sphincter muscles are going to open up so that the urination can occur, a micturition can occur through the urethra. Okay, so now with respect to that, the opening of the ureters, so you can see the opening of the ureters, so they are not straight like this. So it is not completely straight like this. What is happening? So it is somewhat projecting. So why exactly it is projecting? So this will act like a valve. Okay, so valves are basically there to prevent the backflow, or so whenever a certain flow we do not want, so we want to restrict the flow of, so it can be against the gravity also, or it can be towards the gravity also. So any of these cases, if valves are present, so it is basically regulating the flow. Okay, so now they are these projecting ureters. What are they? So they'll act like a wall to prevent the backflow of a urine. So why backflow of a urine occurs? So when this urinary bladder is full, and if the micturition is not happening, so there is a chance that, okay, whenever the bladder contracts, okay, so to pass out the urine, so now the contraction, so now this is the bladder, so this bladder will contract to pass out the urine. So that is when there is a chance that this urine can flow back into the ureter. So in order to prevent that, the ureters are somewhat projecting. Now the sphincter muscles, okay, so you know it is sphincter is a muscle, and you know that the brain always sends a signal through motor neuron to the muscles and the glands. So if you remember that you have studied the nervous system, right? So there you study about brain sending a the impulse or a message through the motor neuron to the effector. So what exactly is effector? It is either the muscle or the gland. So here it is a sphincter muscle which is getting a response from the brain, or an impulse, not the response, impulse from a brain to open up so that the urination can occur through the urethra. So sphincter guards the opening of the bladder into the urethra and relaxes only at the time of the urination or a micturition under the impulse from the brain. So brain is sending the impulse through motor neuron. So through motor neuron it is going to the muscle. So muscle is the effector here. So this sphincter muscle is going to relax, and that is when this particular urinary bladder is going to pour its secretion, basically the urine, into the urethra, through the urethra to the outside. Okay, so that is how the urination or the micturition is done. So let's have a look at the kidney structure here. So I'll just draw it and explain how exactly. So you can see here a longitudinal section of the kidney is taken. Okay, so if we are longitudinally sectioning the kidney, okay, so let's have a look. 1 second, I'll draw the kidney. So this one over here now is a kidney. Okay, so this is a kidney. All right, so kidney is again divided into two parts. So there is actually, so as you can see here, so there is a cortex part and then there is a medulla part. Okay, so you can see in this also. Okay, so this one over here, so I'll draw the cortex part. So let's say here is the cortex part. Okay, and then there is medulla part. Okay, so then you will find a pelvis there. Okay, so let's draw the pelvis here itself as such. Okay, so this is going to form the pelvis. So renal pelvis will be forming, and then later on it is going to be it is becoming a ureter. Okay, so this is now a ureter over here. So I'll just label this one. So this is ureter. Okay, and we know that the kidney is divided into outer part. So this is actually longitudinal section. So you can see here kidney in longitudinal section. So let's say this is a kidney now. Okay, so if we are cutting in this direction, okay, so it is a transverse section. If we are making a cut in this way, okay, so if we are making a cut through the kidney, so in this way, so that we can observe the kidney parts, how it is arrangement is done. So that is a longitudinal section. So when we take a longitudinal section, so you can see a papilla, a cortex region, a medulla, a pyramids, you can see ureter, pelvis, hilum. So we will draw all that and let's see this. Okay, so now here in this case, as you can see, okay, so this one is forming the pelvis region. So let's have a look here. So this cortex region, so cortex region will have a dotted appearance. So why do you think that there is a dotted appearance over here in the cortex region? Yes, anybody? So this is all completely dot like. So when we observe it under a microscope, so this will give us a dotted appearance. But when we observe a medulla region, so there you will find striations. Okay, so striations can be found there. So let's have a look at this one now. So let me just draw. So you can find striations as such. Okay, so lines like this you can see. Okay, so all these lines are forming a end as such. Okay, so again here you will find a striations. So that is basically lines you will find, and end is called as a papilla. So same way here as well. Okay, so there is a striation. So this one is called as a papilla now. All right, so same way over here as well. So let me just say about this one. So here is a papilla again. Here is a renal pyramid, and the end apex of this renal pyramid, what is there? So that is called as a papilla. So let me just label this now. Okay, so as you can see here, so this part is represents, okay, so this is the cortex region. Okay, so this one is the cortex region, which gives a dotted appearance, and this part what you find, so this is the medulla region. Okay, so this one is the medulla region. Now in medulla region, so we can observe the renal pyramids. Okay, so this one is a renal pyramid. Okay, so everywhere you can see that this is basically how exactly this structure is formed. So we will see that. Okay, I'll explain this one. So now this is a renal pyramid over here. Okay, so this is renal pyramid having the papilla. So this apex of the renal pyramid, so this apex is called as a papillae or a papilla. Okay, so I'll just label these now. Okay, so this one is a papilla. Okay, or a papillae. Then this part what you find, so this is actually a renal pyramid. Okay, so this is a renal pyramid, and as you can see, so this whole thing what is there, so this is forming the pelvis. Okay, so this is a renal pelvis, and in the cortex region and medulla region, so we can see millions of nephrons are arranged. Okay, so millions of nephron, or let's say million nephrons. So instead of millions of nephron, because each kidney, when we mention, each kidney will have million nephrons. Okay, so million nephrons are arranged in each kidney, I can write. Okay, so million nephrons. So what is the alternate name which is given for these nephrons? Yes, anybody? So million nephrons are arranged in each kidney. Okay, so these are arranged in each kidney. Okay, so this with respect to this, so this is a part. Okay, so now in the other kidney also will have a million nephrons. So that is how the arrangement of this one is giving a dotted appearance and a line like striations like appearance in the cortex and the medulla region. So let's have a look at this one now. So let's have a look at the picture here. So here you can see one more thing that they have labeled is hilum. Okay, so junction where the pelvis, the pelvis is there, or we can say with respect to the entry of these arteries which can enter, so that we can mention as a hilum. So this junction what you observe here, so that is a hilum. Okay, so let's have a look here. So longitudinal section of the kidney shows two regions. So that is the darker cortex region and inner the lighter medulla. Okay. So outer cortex region, what is there? So that is somewhat darker as compared to the inner region medulla, which is lighter. Okay, so medulla is composed of finely striped substances arranged in several conical pyramids. Okay, so that I have labeled as a renal pyramid, and the apex of each pyramid is papilla. Okay, so this projects into the pelvis of the kidney. So kidney is composed of enormous number, that means many number of uriniferous tubules. Know the alternate names here. Okay, so you need to know all the alternate names. So I just wrote here nephron, I wrote. Okay, so don't think that only you can use nephron here. So they can ask you uriniferous tubule. So write a structure of a uriniferous tubule, or show how exactly these uriniferous tubules are arranged in the kidney, or nephrons, or renal tubule, just kidney tubules. So any of these names they can ask. So do remember every alternate name which is given for one particular term. Okay, so these are structural and functional units of a kidney. Okay, so here they have shown the blood supply also. So renal artery, so this one is the artery, so renal artery is coming. Okay, so this will be a dirty blood. So why do we call it as a dirty blood? Because it is anyway oxygenated blood. There is a waste product which is carried by this one. Okay, so this waste product has to be got rid of. That is why it is coming to the kidney. So now this waste product is removed by the kidney, and then the pure pure blood is leaving. Okay, so that is not a pure blood, like it is not a waste, so it is not having a waste product as such. So that will be leaving through the cleaned vein. So that is a renal vein, that is a cleaned blood will be leaving. So this one is a pelvis over here. Okay, and then the ureter formation is there. So I hope it is clear. So I'll just draw a structure of a nephron and show you how exactly the arrangement is done. So let me just explain this one. Okay, over here. Okay, so you know the structure of kidney tubule, right? So this one second. Where is it? Okay, so now as you can see, okay, so there is a kidney tubule. So let's draw the Bowman's capsule. So this you need to know, and this itself, the whole structure, the same structure is actually arranged in the kidney. So let's have a look. So this is now a Bowman's capsule, a cup shaped structure, what you find here. Okay, so this is a cup shaped structure. So you can see there is a proximal convoluted tubule. Okay, so this one is a proximal convoluted tubule. So this one is forming a descending limb, then there is ascending limb. So this is a descending limb. Okay, then there is ascending limb which is there. Okay, so this one is a ascending limb. So again there is a formation of convoluted tubules here, and then it is joining where? It is joining the collecting duct. Okay, so now here you can see a collecting duct which is present and is responsible for urine collection from different parts. Okay, so this is now a collecting duct which is there. Okay, so let me just label this one. So this is now a Bowman's capsule. Okay, so this is a Bowman's capsule. So Bowman's capsule, one second. Okay, so this part what you observe here, so this is a Bowman's capsule. Okay, so Bowman's capsule along with the glomerulus. So they'll ask you to draw a structure of a Malpighian capsule. Okay, so you need to draw the Malpighian capsule also. Let me just draw that as well here itself. So there is a afferent arteriole which is coming up here. Okay, so this will further divide, and there is a formation of efferent arteriole which we can see. So capillary formation we can see here. So a ball of mass, a knot like mass formation we can see over here, and you can observe here with respect to the diagram itself. Okay, so the diameter of the efferent arteriole. Okay, so this is the efferent arteriole, and this one is the afferent arteriole. Okay, so here this one is wider, the radius, the diameter is wider, and here the lumen is narrow. Okay, so wider lumen we can find and a narrow lumen we can find. So that is basically, so difference in this itself is creating a hydrostatic pressure over here. Okay, so there is a hydrostatic pressure. Okay, so let me just write here. So this one is hydrostatic pressure is created, and that is the reason why ultrafiltration process occurs. Okay, so now in this case, just a second. Okay, so you can see here this one is actually a glomerulus part. Okay, so this is a glomerulus part. So I'll just take this one also for labeling now. So glomerulus and along with the Bowman's capsule. Okay, so these two together, so they are glomerulus. Okay, so these two together are forming the Malpighian capsule. Okay, so this is a Malpighian capsule. Okay, so they'll ask you to draw the structure of a Malpighian capsule. Make sure that you are drawing the wider lumen over here as compared to the other one. So narrow lumen is here, and here the wider lumen is there. Okay, And let's label each one of these now. So labeling for this one goes as proximal convoluted tubule. This is PCT. This is distal convoluted tubule. This one is Henle's loop. Okay, so this is Henle's loop, and this is actually the collecting duct. Okay, so this one is a collecting duct. So now in this case we need to see how exactly this structure is arranged in the kidney. Okay, so you know that we already discussed about this one. So let's have a look now. 1 second. So I'll just make a partition in that so that you can understand how exactly the arrangement is done. So this part what you find till here. Okay, so this whole part what is there, so this is the part which you can find in the cortex region, and this below part, that is Henle's loop and the collecting region, collecting duct. So this you will find in the medulla region. Okay, so these are found in the medullary region. So that is why you see that renal pyramid, right? So this one is a renal pyramid. So renal pyramid formation is mainly because it consists of the collecting duct. Okay, so this will be made of collecting duct, and with that a Henle's loop is also there. So Henle's loop and collecting duct are present in the renal pyramid, and that is why you can find a striations there, or stripes like this. Okay, and this one is giving us a dotted appearance. Okay, so this dotted appearance is mainly because of PCT, DCT, and also the Bowman's capsule. Okay, so all these three we can find in here, and that is how we are getting a dotted appearance when observed by taking a longitudinal section. So here this is the structure, and this particular capillary what is there, so this will further divide. Okay, so now afferent arteriole is there, efferent arteriole is there. So as the volume is less here, as the diameter is very narrow, what will happen? The volume is actually decreasing, the pressure is increasing. So as the pressure increases here at this side because of the narrow diameter over here itself, so what is gonna happen? So there is ultrafiltration process is gonna occur. Okay, so hydrostatic pressure is generated. Okay, and because of this pressure itself, the filtration of a blood will occur. So all that filtered liquid what is there, so that will be entering this glomerulus, and that we call as a glomerular filtrate, and it will start moving down. Okay, so this is with respect to structure. So let's have a look at this one. Yes, are you all understanding? Okay, I don't understand why there is okay, so there is actually chat is not visible. So this happens for me every time. Need to get it once, like checked once. All right, so yes, okay, 1 second. Yeah, so each kidney tubule has the following parts. So now we need to discuss the blood supply. Okay, so how exactly blood supply? So here I was drawing the blood supply is done through the kidney. So let's have a look now. Okay, so as you already know that, 1 second. Yeah, so as you already know that each of these kidneys what are there. Okay, so here I had drawn a kidney, right? So for them there will be a blood supply which will be done. So that is the dirty blood is coming over here and it is joining. So this one is actually the renal artery. Okay, then what is happening after the filtration is done? So renal vein is leaving back. Okay, so renal vein is going back to the heart. So that is the cleaned blood we can find as the filtration process has occurred here. So now with respect to that, so let's have a structure of kidney here. So this is a kidney. Okay, as the renal artery comes, okay, so as the renal artery comes, so you know that there are millions of nephrons here. Okay, so millions of nephrons are present in this case. So what is happening? So this renal artery has to divide. So if I I have to discuss this one. So let's have a look this one second. I'll just take the different page. Yeah, so now the artery will divide. Okay, so artery will divide and it will divide to form a arteriole. Okay, so this is a arteriole. So this is now a artery which is carrying a dirty blood to the kidney. Okay, then it is dividing to form a arteriole. And where do you find a arteriole? Arterioles you will find at the kidneys, that is the nephrons mainly. Okay, so afferent arteriole and efferent arteriole. So this artery, since there are millions of nephrons are present in a kidney, okay, so this same artery has to branch million times. Okay, so this has to branch million times so that it is providing a arteriole to all the different types of all the different nephrons which are present. So all the different nephrons are getting this arteriole. So to make sure that so arteriole which is there, so artery is dividing to form a arteriole. So let's have a look now. So now the arteriole will divide, and from arteriole itself further division we can see, and this is going to form a capillary. Okay, so capillary formation we can see here. Okay, so after that what will happen? The efferent arteriole formation occurs. So that we call, so here in the normal cases we will say venule formation, and then venules will join together, vein formation. So let me just complete that. Okay, so after through the capillary, once the exchange occurs, okay, so that is gaseous exchange is gonna occur through this part. So as the gaseous exchange, the waste product is given out, that is filtration is occurring. So now the venules are there. Okay, so these venules will join together. Okay, so there is a venule joining, and then there is vein formation which occurs. So this is now a renal vein which is joining the heart. I mean, it is going to the heart, and then so most of the things like exchange of gases which was done. So same way deoxygenated blood, clean blood is there. So why do we call it as a clean blood? Because waste product is anyway removed, but again it is a deoxygenated blood with respect to carbon dioxide. So that has to be anyway purified again. So that is why it is vein which is actually carrying a deoxygenated blood itself, but again cleaned blood we are saying because it is not having a nitrogenous waste, because artery was carrying a nitrogenous waste, and purification of this nitrogenous waste, like removal of the nitrogenous waste was done through this arteriole, that is afferent arteriole, which was coming into this particular Bowman's capsule. Capillary formation occurred, and then efferent arteriole. So let's have a look here. Okay, so in this case, so this artery itself will divide million times, and again after all the filtration is done, so this is joining all those efferent arterioles which were there. So they are all joining back, and there is vein formation which is occurring. Okay, so here what exactly is happening? The arteries are, the artery which is coming, so this is branching. Okay, so branching is occurring and rebranching is occurring. So joining is also occurring. So branching, rebranching, joining is occurring, so that millions of nephrons which are there in each kidney, so each and every nephron has to get a arteriole. Okay, so once the arteriole is received by each and every nephron, then here the filtration process will occur, then it is leaving. So let's have a look at this one now. So as it leaves here, so it is not just like this. So what is happening here? So this is further forming the capillaries around here. Okay, so there are secondary set of capillaries that are formed as the reabsorption process will occur here. Okay, again what is happening? So again there is a formation of capillaries around this structure. So there is a formation of capillary around this structure. So this is joining again. Then there is again a formation of capillary around this structure as well. So only after this, so here itself it is not a vein actually. Okay, so here it is still a efferent arteriole. It will further divide, it will form the secondary set of capillaries here. Okay, so secondary set of capillary formation, why it is occurring? So this is occurring mainly for the reabsorption, because as the filtration occurs, so we don't want, I mean our body doesn't want to get rid of all the water content, so some of the water content is still useful, the glucose which is there, so that is also still useful, and so some salts are there, so that might also be required. Okay, so basically because of the pressure, most of the liquid part will get filtered here, but it is not always, and it is not urine, it is not at all completely a urine. So only after it crosses the DCT we can call it as a urine. Until then it is a glomerular filtrate itself. So this part what we are getting, the liquid, so that is actually a glomerular, okay, one second, this is not writing, yeah, so that is actually a glomerular filtrate. Okay, so after the glomerular filtrate is reaching, so as the secondary set of capillary formation occurring, so here what happens, reabsorption process occurs, then here at this point tubular secretion occurs, and as all these processes are completed, so now all of this, so there is a gaseous exchange also which is occurring, so all of this will join together. So I need to draw this in blue color so that you can understand here, as it goes till here. Okay, so from here itself I need to make it a blue color because a gaseous exchange also has taken place. That means oxygen supply has been done to the tissues of the nephron. Okay, and carbon dioxide which is formed in these tissues, so that is collected. So that is why the blood which is leaving, that is a cleaned blood, but it is a deoxygenated blood. So now all these capillaries will join and there is a venule formation. So from different of these, so there is a venule formation and then there is a vein formation. So this is now a renal vein which is going out of the kidney. So other nephrons what are there, so from there also capillary joining is occurring, from others also there is a formation of a capillary. So here we have drawn a collecting duct, right? So it is not just for one nephron, so many such nephrons are creating or pouring their contents there. So that is basically a urine. So urine is poured from different nephrons which are interconnected here through this collecting duct. So once the process, that is once the DCT, once it crosses the DCT, so then only we can say that the urine formation has taken place. Okay, so I hope it is clear for you all. I can't even read your comments, so that's the reason I'm not able to respond to you. If you have written anything, so you can get in touch with me on special classes as well. Yeah, one more thing I want to say. So I'm conducting now itself at 5:15, so soon after this session, special class I'm conducting on circulatory system, that is a pole war I'm conducting. Okay, so pole war on circulatory system. Okay, so we will solve about, I have added about 55 questions. So 55 questions I have added, so let's solve this. So those for whom it is possible to join the class, so all you need to do is download the Unacademy Learners app and select your category, like enter your mobile number, email ID, everything you are doing, and after that enter, you can enter the category ICSE class 10. Then you can find me, so you will find my class. So you can just search for my name Vani Kari. Okay, so you can search for my name Vani Kari, so you will find my special class there, so you can take up this quiz as well. Okay, so this is gonna help you because all important questions I have added there. So this pole war you can attend at 5:15 p.m. Yeah, I was saying about this one. So since the chat is not visible, okay, I'm not able to respond to you, so you can get in touch with me on my Telegram channel as well. Okay, so now I was saying about this one. So let's have a look at this one. The Bowman's capsule, it is a thin walled single cell epithelium. So this is the Bowman's capsule now. So why do we call it as a thin walled single cell epithelium? Because this is the structure. So only one line of epithelium is there. Okay, so there are no multiple layers, there are no multiple cells which are present. So one layer of a cell is there. So it is not, the nephron is not like one whole cell actually. So this is made up of many cells, okay, but you know a layer of cells are there. So now this is many cells are there but not many layers, okay. So one layer, so single layer of epithelium we can find. So Bowman's capsule, this is thin walled single cell thick epithelium. So there is no compound epithelium, there is a simple epithelium. So this is a cup, something like a hollow ball pressed deep down on one side. So if you press a ball only from one side, so you will get a cup shape, okay. So that is like a half moon shape also we can mention. So its hollow internal space continues into the tubule, okay. So the outer concavity of the cup lodges a knot like mass. So this we saw the blood capillary which is being formed from the afferent arteriole, which is continuously dividing, and by dividing what is happening, so here I was saying, so this afferent arteriole, okay, so afferent arteriole will divide further and there is a formation of this capillary. So this itself is a glomerulus, okay. So glomerulus formation is occurring. So this Bowman's capsule and the glomerulus together, so both of them together, so we call it as a Malpighian capsule or just the renal capsule. Again, know the alternate names which are mentioned. So here I have drawn Malpighian capsule, I have written. So renal capsule also can be mentioned for this one, okay. So if they ask you to draw a structure of a renal capsule, don't keep on thinking like what exactly it is, okay. So if they are asking you to draw a renal capsule, just draw renal capsule. You need not draw PCT, Henle's loop, DCT, collecting duct and other things. Just a second, I'll plug in the laptop. Yeah. So what happens is, so only you need to draw that. So you, like, you know, if you are drawing extra, so they'll think, so evaluation with respect to IC is done that manner. So if you are drawing anything extra, so you know that renal capsule is just the Malpighian capsule, not the whole kidney tubule, like not whole nephron. But if you are drawing the whole structure, then they think that you don't know what exactly is renal capsule, okay. So you don't know exactly what exactly is Malpighian capsule, so that is why you are drawing the whole structure. So that is what they'll think and you will lose marks. So whatever they have asked, you write only that. Whatever they have asked, like, you know, they have asked you to draw Malpighian capsule, what you are doing, so you will draw only the Malpighian capsule. So now this is the Malpighian capsule, okay. So this is Bowman's capsule and here the afferent arteriole is there. This one is a glomerulus now, okay, a set of capillary, efferent arteriole. So only this much you need to draw and the labeling has to be done. Afferent arteriole, efferent arteriole, glomerulus, okay. This is Bowman's capsule, both of them together Malpighian capsule or a renal capsule. So this if you draw, that is more than sufficient. And one more thing you need to keep in mind, so this was your sample paper. In one of the sample papers, so they had asked to draw, so just the sample paper I'm discussing, okay. So they are asked to draw the structure of this, I mean Malpighian capsule. One student, so he or she, I don't know, so they did not make the differences here. You know, the wider lumen will be here, the diameter is wider here and the diameter in the efferent arteriole, so this has to be very less. So when you are drawing a Malpighian capsule, make sure that you are very careful with respect to each and every detail, okay. So this has to be wider, this has to be narrower, because so only if a narrower area is there, that means volume is less there. So you know that volume and pressure are inversely proportional. So if the volume is less, the pressure is going to increase, okay. And as the pressure is increasing, the hydrostatic pressure is more. As the hydrostatic pressure is more, the filtration of a blood can occur. So you need to be very careful while you are drawing this one. So just to check also whether you know that would the diagram with respect to diameter or wider lumen, narrower lumen. So they'll ask you all of this, so please be careful about such things. So concentrate on each and every detail. Okay, so you will do well about your exam, and with respect to your exam, so you will do well, but be careful with respect to each and every details. So the give reasons which are mentioned, so when give reasons are asked also, you need to be very careful with respect to such things. Okay, so yes, I was saying about this, so this we discussed already. Okay, so proximal convoluted tubule is basically the starting convoluted region of the tubule itself. So why do we call it as a proximal convoluted tubule? Because this convoluted tubule, what is there? Okay, so this is nearer. Proximal meaning is PCT. Proximal is nearer. Okay, so what is nearer here? This tubule. Okay, so this convoluted tubule, what is there? So this is nearer to Bowman's capsule. Okay, so since it is nearer to the Bowman's capsule, that is why the name is given as a PCT. And you can see here, so here the Henle's loop will continue, and then DCT is here. Okay, but as compared to the PCT, so here this one is named as a DCT. That means distal convoluted tubule. This is distant from what? Distant from the Bowman's capsule, because it is at a distant from the Bowman's capsule. That is why it is named as a distant convoluted tubule. So each and every details, if you remember, so you can clearly write your diagram, draw your diagrams, write the reasons also why it is like that, why it is named as a distant convoluted tubule. Okay, so here the proximal one, the proximal means nearer. Both the Bowman's capsule and the proximal convoluted tubule, so they are all lying in the this part. So you can see here, so this Bowman's capsule, PCT, DCT, so they are in the cortex region of the kidney. Okay, and the other one, so that is the Henle's loop and the collecting duct, so these are there in the kidney medulla region. Okay, so that is why the pyramid shape we can see in the medulla region, that is striations we can see, and this one in the cortex region, so we can see it as a dotted appearance. Okay, so before I end the class, I hope this was all clear to you. So this is the structure that we have discussed, and the time is also up. Okay, so see you all in the next session, and if possible for those who are able to attend this session, so if it is possible for you to attend the session, take up the quiz on circulatory system, so you can join in the class soon after this class. Now at 5:15 I'm having. Okay, so yes, so see you all in the next session. So before that, let me just anyway, even if I ask you a question over here, since I cannot see a chat, so this is of no use, I'll just answer this one. So another name for a renal tubule is, it is the uriniferous tubule. So every alternate name you need to know. Okay, so other than that, the Bowman's capsule and the glomerulus together, so this is called as a Malpighian capsule or a renal capsule as well. Okay, so reuniting capillaries of the glomerulus form. So now afferent arteriole was there is a capillary formation. Okay, so now all of them are reuniting, so this reuniting capillary is efferent arteriole. Okay, so reuniting capillary, all the capillaries which are here, so they are all reuniting and efferent arteriole is being formed. So this one was the afferent arteriole, this is the efferent arteriole. So opening of sphincter muscles for micturition is by the impulse from the brain. So whether it is true or false, yes, this is true, because under the impulse from the brain itself, the relaxation of a sphincter muscle will occur, and then the urination is gonna occur. Okay, so before I end the session, so let me just say about this. So there is Udan batch and Champions batch. So these are ongoing batch. You can have, you can watch the recordings also if you are going for the subscription. So this is for plus subscribers, So if you are planning to take a subscription, so you can use my referral code VANIYT to get 10% discount on the subscription. Okay, and the mode of delivery in this batch is Hindi, the mode of delivery in this batch is basically English. So you can go for any of these batch, and we are conducting one short revision. We are also conducting a poll war, so you can take up this session as these are going to help you crack your finals. Also we are conducting a test series for plus learners. Okay, so basically it is 60 minutes and 30 questions will be there. So again this is gonna help you crack your finals. Okay, so you can take up this as well. So you get one more chance for prodigy, so that is see you on Feb 27, that is 12 to 1 p.m. So you can take a, you can spend 60 minutes of your time, so take up this aptitude test. So there is a chance that you might win this particular scholarship, the college grant. Okay, that is the scholarship. And also you can win the iPhone, MacBook Air, Amazon Echo Dot, Amazon Watcher. So any of these you can win. So there are different slabs are available for scholarship also. So you can enroll now for this one, and to unlock you can use the same code VANIYT. Okay, so once you download the Unacademy Learner app, so you can get this link, or you will find this in the description box also. And after that what you can do is, so it will ask you unlock code, so you can use my referral code VANIYT to unlock. So download the Unacademy app now and try all of these features for free, and this is my referral code. So if you are taking a subscription, so use this referral code, so you will get 10% off on the subscription. Okay, so how to watch my special classes? So all you need to do is download the Unacademy Learner app, and then open the app and click on get started. Enter your mobile number and OTP, sign up using your email ID, then select class 6 to 12. Then you can choose your category ICSE. Okay, and after that you can choose the language that you want to learn in. Okay, then to unlock the free product, so you can use the code VANIYT. So wherever it asks you code, so you can use the same code to unlock. Okay, so this one, you can subscribe to us on Unacademy ICSE and ISC boards, the YouTube channel, and download the Unacademy Learners app. So see you all in the next session. Bye bye everyone, thank you so much.